Hi everyone, my name is Raghav Herugu, and today I would like to talk about artificial intelligence for oral squamous cell carcinoma detection. Next slide, please. So OSCC, or what we like to call oral squamous cell carcinoma, involves a significant degree of damage to what we refer to as epithelial cells, which form the protective layering of surfaces such as our skin and indeed the insides of our mouths. These cells have a vital role in our health, and it is their impairment that can lead to the onset of this form of cancer. It is important to note that the prevalence of OSCC in the global cancer statistics is alarming, which underscores the urgency of our understanding, awareness, and measures for early detection and effective treatment strategies. As we delve deeper into our discussion, we will be exploring more about the nature of this disease, its causes, symptoms, and potential preventative measures so we can better understand its overall impact on global health. Next slide, please. The primary focus of our exploration today is centered around the application of artificial intelligence, or AI, in detecting oral squamous cell carcinoma. The spotlight in particular is on a branch of AI known as deep learning. Imagine deep learning as a very meticulous student who learns by observing patterns and examples. Over time, the student becomes extremely proficient at identifying patterns, even in complex data such as medical images. This proficiency is what we aim to harness in our fight against OSCC. Let's delve deeper into the heart of this technology that powers this meticulous student we call deep learning. And this is where neural networks come into play. Imagine a neural network as a vast web of internet-connected nodes or neurons that mimics the structure and function of the human brain. Each neuron is like a tiny processor that makes calculations and passes information to the next layer of neurons, similar to how our brain cells pass signals to each other. In the context of our study, these neural networks have a very important job. They take the input, in our case, the images of tissue samples, and process it through multiple layers of these interconnected neurons. Each layer extracts and learns different features from the images. The first layer might recognize simple patterns such as lines, edges, and things like that. And as the information goes deeper, the patterns recognized becomes more complex, allowing the model to identify intricate structural characteristics of OSCC. In our study, we deployed these neural networks utilizing their remarkable pattern recognition ability to analyze the tissue samples. By training the neural network with a large number of these images, we helped it learn to differentiate between healthy and cancerous tissues, honing its proficiency in detecting OSCC. In essence, neural networks stand at the core of our AI model's ability to learn and make sense of complex data, providing us a powerful ally in our battle against cancer. In our study, we've delved deeper into the inner workings of these deep learning models. Our focus has been on exploring the varying impact levels of two key techniques, regularization and dropout layers. These techniques are the fine-tuning knobs of our AI models, helping us to prevent overfitting, where the model performs well in training data but struggles with new unseen data. This exploration brings fresh insight into the field, adding to the body of knowledge in this area. It is akin to a new chapter in an evolving textbook, providing a deeper understanding and perspective. For those interested in further reading, there are similar papers that also explore this exciting frontier. These include a study published on PubMed titled Automatic Detection of Oral Squamous Cell Carcinoma from Histopathological Images of Oral Mucosa using Deep Convolutional Neural Network, and another paper with a similar focus published on DPI. Next slide, please. The focal point of the research that we're going to discuss today is an exciting and potentially transformative one. We're venturing into the realm of harnessing the power of artificial intelligence, or AI, to combat a serious health concern, OSCC. Our journey of exploration centers around the intriguing question: can we train AI models to become adept at detecting OSCC? And we're not asking our AI to make this detection based on simple data. Instead, we are challenging it to analyze complex images of tissue samples, a task that requires a sophisticated level of understanding and pattern recognition. To put it in simpler terms, imagine you're trying to find a specific pattern in a complex tapestry. This is the challenge we're proposing to our AI models. Our objective is to guide these models in learning to discern the subtle yet crucial differences between healthy tissue and tissues affected by the oral squamous cell carcinoma. This research paper is essentially a chronicle of our journey and exploration of this intriguing possibility. It is an exciting venture that could potentially revolutionize how we approach OSCC detection, and in doing so, significantly enhance our ability to fight cancer. Next slide, please. So I'd now like to share with you a glimpse of the very raw material that powered our AI models, the microscope images used in the study. These images, magnified 400 times their original size, served as a textbook from which our AI model learns. On the screen, you can see two such microscopic images. The image on the left represents a cancer-free or healthy tissue sample, while the image on the right represents a sample of tissue afflicted with the cancer. In our study, we fed our AI models a considerable number of such images, 36,410 cancer-free images to be precise, and a slightly lower number of 35,497 of the oral squamous cell carcinoma images. Think of it as teaching a child to distinguish two types of fruits by showing them thousands of examples of each, which each image, the model learns a bit more, fine-tuning its understanding and sharpening its ability to distinguish between healthy and cancerous tissue. The task of distinguishing between these seemingly similar images is not easy, but it is crucial. Through this, we aim to empower our AI model to accurately detect OSCC, ultimately assisting medical professionals in making accurate diagnoses and potentially saving lives. Next slide, please. Initially, the data set we obtained from an online source consisted of just 201 normal oral cavity histopathological, or NOCH images, and 495 of the cancer images. To make our study robust and our model learning comprehensive, we needed a much larger pool of images, and that's where the magic of data augmentation came into play. Think of data augmentation as a creative artist who can take a handful of pictures and transform them into a diverse gallery of images. Using data augmentation techniques such as random rotations, flips, and other transformations, we were able to significantly enrich our data set. We essentially augmented our initial pool of images of 201 and 495 to a staggering total of around 35,000 images for each. This vast and varied data set allowed our AI model to learn from the multitude of scenarios, enhancing its ability to identify OSCC in a wide range of situations. This, ladies and gentlemen, is the power of data augmentation and AI, enabling us to make the most of our available resources and contribute against the fight against cancer. Next slide, please. Imagine you're trying to teach a child to understand the concept of a bird. You show them pictures of sparrows, eagles, and parrots, and they start to get the idea of what a bird is. But then they see a picture of a penguin and they get confused. It doesn't look like the other birds. If the child insists that a bird must always have wings or the bird must always be flying, they've overgeneralized the concept, similar to how an overfitting machine learning model might behave. Regularization is a technique where we use to prevent this overfitting, the scenario where a model will so precisely fit to the training data that it fails to generalize well to unseen data. In essence, regularization helps us maintain a balance. It prevents our model from becoming an overachiever that performs exceptionally well on training data, but fails miserably when faced with new unseen data. Now let's talk about a specific regularization technique known as dropout. Dropout in machine learning terms is a form of temporary amnesia for our model. During the training process, dropout randomly forgets or drops out a fraction of the neurons in a neural network layer. This step is done to ensure that the model does not rely too heavily on any single neuron, thereby promoting a more robust and generalized model. Think of a dropout as a team building exercise. If one team member takes on all the tasks, the rest of the team may not learn or contribute as effectively. But if the tasks are distributed evenly, the team becomes more resilient and adaptable, just as our model does with the application of dropout. Next slide, please. Let's now delve into the specifics of our experimental setup, and I'll try to break it down in a way that some of us who are not familiar with machine learning can understand. Firstly, our test data set. This is a collection of images that we use to evaluate how well our model performs. Think of it as the final exam for a machine learning model. In our case, the exam included 24 images of NOCH, or normal images, and 50 images of OSCC. When we talk about accuracy, we're referring to how often our model gets the predictions right. And when we're discussing loss, we're looking at how far off our model's predictions are from the actual values. In essence, we want the accuracy to be high and the loss to be low, and this would indicate a successful model. Each model ran for what we call 100 epochs. An epoch is simply one full cycle where the model has seen all the training data once. In our case, the 100 epochs took 17.5 hours on my computer, a testament to the complex nature of the task at hand. We plotted what we call loss and accuracy curves. These are graphs that help us visually understand how our model's performance changes over time. Think of it as a progress report card that we use to track how a student is doing over a school year. Next, we come into the validation data set. This is like a pop quiz for our model during its training. We use 75% of the available images for this purpose. These regular checks will help us understand how a model might perform on the final test, allowing us to make tweaks and improvements along the way. Finally, we use a batch size of 32 for these models. The batch size refers to the number of samples that the model looks at before it makes an update to what it has learned. It is like reading a few pages of a textbook and then updating your understanding before moving on to the next few pages. Next slide, please. I would now like to guide you through some important visuals over the course of our that depict the performance of our machine learning models over the course of our training. These visuals are represented in the form of 4 curves: training accuracy, training loss, validation accuracy, and validation loss. To make this easy to understand, imagine these curves as a snapshot of a marathon runner's performance over the course of the race. The goal is not to just finish the race, but to do it in an optimal way. Let's first take a look at the top left graph. This one represents our first model's validation loss in green, which is much like tracking a runner's fatigue over time. We can see that the model goes through the 100 epochs or rounds of learning. The validation loss slowly increases. This is akin to a runner getting progressively more tired as the race goes on, and this is not exactly what we want. Now, if we go to the top right graph, this one tells a different story. Our second model's validation loss gradually decreases as it progresses through the 100 epochs. This is equivalent to our runner pacing itself better, maintaining their energy, and getting less fatigued as the race progresses, a far more desirable situation. Now let's drop down to the bottom graph. This represents our third model. Here we can see the validation loss decreases similar to the second model, but the overall loss is lower than the second model. And this is like a third runner who not only paces themselves during the race, but also finishes with less fatigue than the second runner. However, it is important to note that if the second model, our second runner, were allowed to continue the race for a longer duration, it shows promise that it could eventually surpass the third model in terms of lower loss, as it had the lowest test loss. This is akin to a runner who, given more time, could potentially outperform the others due to their superior racing endurance. Next slide, please. Okay. Let's delve into some numbers that illustrate this. When we employed L2 regularization along with 2 dropout layers set at 0.5, our model achieved an impressive accuracy of 89.19%. To put it in perspective, it's like our model correctly answering nearly 9 out of 10 questions on a test. On the other hand, when we did not use any regularization or dropout techniques, the accuracy dipped down to 81.08%, akin to getting just over 8 out of 10 questions correct. Similarly, when we use L2 regularization with 2 dropout layers set at a higher rate of 0.8, the accuracy was 85.14%. These findings underscore the profound impact of regularization and dropout techniques in improving the accuracy and reliability of AI medical image analysis. They help to address a common problem known as overfitting, where a model performs exceptionally well on training data, but does not do so well on unseen data. This is akin to a student who does well on practice tests, but struggles in the real exam. By enhancing the ability of our model to generalize from training data to new data, these techniques significantly improve cancer detection capabilities, moving us one step closer to leveraging AI's full potential in the fight against cancer. Next slide. As we reach the concluding part of our discussion, I'd like to cast your gaze forward and explore 2 potential avenues for future research that can build upon the foundation we've laid in our study. Firstly, we have the opportunity to delve deeper into the fine-tuning of what we call hyperparameters for regularization and dropout techniques. Hyperparameters in layman's terms are like the adjustable knobs on a machine. By tweaking them, we can significantly impact the machine learning's performance. By optimizing these knobs in the context of our AI models, we can believe that we can further boost accuracy in detecting medical conditions. Our second direction is to look beyond the realm of oral squamous cell carcinoma. We envision applying these AI techniques to detection of other types of cancer, and the potential impact of AI in medical image analysis is truly phenomenal. In conclusion, while we've made promising strides in this study, our journey is far from over. The future holds exciting possibilities for AI in the realm of medical diagnostics, and we're just beginning to tap its full potential. Promise of future where AI significantly contributes to our fight against cancer is indeed a bright one. Thank you for listening.